Hey everybody, thanks for joining me again. I want to take a moment today and talk about something I'm really excited about. I like to geek out in biochemistry and cell biology, and one of my favorite areas to research right now is the field of exosomes. Now, you can call exosomes, they're called by so many different names. You can call them nanoparticles, nanospheres, extracellular vesicles, and none of that nomenclature would be incorrect. But what these are structures that are formed by a budding from the plasma membrane and also from the endoplasmic reticulum, but that's a story for another time, that capture cytosolic components in a various cell. And what happens is these continue to coalesce and coalesce and coalesce into larger multivesicular bodies is what they're called, and then these large compartments of endosomes and exosomes is then released into the extracellular space. But what's most important about these are the messages, what's trapped inside that vesicle. And the contents of this vesicle vary from cell type to cell type and really reflect what that particular cell does in nature. So for example, exosomes from mesenchymal stem cells are going to look different than exosomes that might be derived from hepatocytes in the liver, right? So that makeup is going to be a little bit different. But in general, what you see with these, another way to look at these exosomes, they're largely involved in communication. They allow cells to communicate with other cells nearby and also with other cells at a distance, all in the spirit of regulation. Now, the contents of these cells, largely full of cytokines, chemokines, regulatory RNA, also called microRNA, some messenger RNA, some lipids, and various other factors that communicate, that can, when taken up by a nearby cell or a cell that's at a great distance, can change, can upregulate or downregulate various gene expression and ultimately, various protein phenotypes for that cell and change the behavior for that cell. When you're talking about cytokines and chemokines, those tend to be very immunomodulatory. So you're always going to have that element, when you're talking about what's in an exosome. So I mentioned cellular communication, and there are generally three modes. There's autocrine, paracrine, and endocrine. Autocrine would be if a cell releases exosomes and then those same exosomes act upon that very same cell. That probably does happen, but that's probably a minority percentage of the mode of communication of exosomes. Paracrine means these exosomes released from a cell are going to act on nearby cells. Those cells could be in the same tissue, for example, or nearby tissues. And then finally you have an endocrine, which probably a lot of us are familiar with, the field of endocrinology and hormones and such, where these exosomes can act at a great distance. For example, if you had cells from your adrenal glands releasing exosomes that move in the bloodstream to distant part of the bodies to affect different things at different functions and elicit different responses for various cells. So what's most interesting to me, I come from a pharmaceutical chemistry background and drug delivery design, things like that. So the ability to engineer exosomes and load exosomes with particular messages to counteract pathologies or to correct aberrant states, to me that's really interesting. But what we're going to do going forward is we're going to spend a great deal of time talking about the potential therapeutic benefits of these vesicles, particularly those vesicles that arise from mesenchymal stem cells. So this was just a surface view of exosomes. We're going to take deeper dives as we move forward. Thanks for joining me today. Stay regenerative, my friends.